Mm, just keep in mind. Your screen.
जमलेल्या माझ्या तमाम हिंदू बांधवांनो भगिनींनो आणि मातांनो
रिश्ते में तो हम तुम्हारे बाप होते हैं नाम है शहंशाह
Police Station, Churchgate. station, Churchgate.
Mm.
Hello, sir.
Hello. Hello. हॅलो सर.
Hello.
हां सर, अनाऊन्स करा सर तुमचं.
हॅलो सर.
हां करा सर अनाऊन्समेंट.
लाईव्ह नाही ना आपण अजून?
हो लाईव्ह सर 9 वाजता. बोला लाईव्ह. सर वी आर लाईव्ह नाऊ. हॅलो.
Okay. Good morning, one and all. Welcome to the session 3 on day 2. Due to the overwhelming response for presentations, there is slight change in the presentation, the schedule. Now we are beginning at 9 a.m. for oral and first section of oral and poster presentation. So now the schedule of presentation is already been mailed and posted on the participants group. Now I'll quickly read the rules or instructions for oral and poster presentation. Presentation, as you all know, shall be conducted on the WebEx platform. The link for the WebEx will be was emailed to the participant on their registered email ID. All participants are instructed to log in for their presentation and share the presentation as per their schedule. Now, rules for poster presentations. Participants will get maximum 5 minutes for explanation. A reminder will be given after 4 minutes by the judges. Participants are requested to share their presentation on WebEx. Now, rules for oral presentation. Participants will get maximum 8 minutes for explanation and a reminder will be given after 6 minutes by the judges. And again, for oral presentation, participants are requested to share their presentation on WebEx. In case, if participants, those who have already submitted their poster and oral presentations, and they are finding any problem for uploading their presentation will be uploaded from our end. Okay. So those were the rules for poster and oral presentations as per schedule. Poster participants for poster presentation P1 to P11 and P12 to P21. The presentation will start at 9 a.m. and they will end at 10:30 a.m. So from 9 a.m. to 10:30 a.m. poster number P1 to P21 will make their presentation. And oral presentation O1 to O14 will join their respective group and their timings are 9 a.m. to 10:30 a.m. For poster presentation P22 to P50 their presentation is scheduled in post-lunch session starting from 3 p.m. and for oral presentation from 0:15 to 0:30. The presentation is scheduled in post-lunch session in their respective group from 3 p.m. onwards. So my best wishes for participants, and participants now can join their respective group, and I request judges to start their presentation as per schedule. Thank you.
A very good morning to one and all. We've already started our day, the second day of the conference, International Conference on Emerging Methodologies in Pharma, Environmental and Life Sciences. A session of oral and poster presentation has just been completed, and moving to the further part of this session, the next session, and we have with us Professor Gulab Khedkar, who will talk on small stories, big impacts, DNA in life science research. I am extremely happy to welcome Sir and at the same time thank him for accepting our invitation. To begin with, let me introduce you to Professor Gulab Khedkar. Professor Gulab Khedkar is director of Paul Herbert Center for DNA Barcoding and Biodiversity Studies in Dr. Babasaheb Ambedkar, Marathwada University, Aurangabad in India. His expertise is in the field of genomics. Simultaneously, he is exploring population genetics for various population groups. Majority of the research tools include Illumina sequencing, genome annotation, mitochondrial genomics, transcriptomics. His research group has aided in resolving various taxonomic ambiguities, often related to several taxa and correlated to evolutionary histories. Sir has published several research articles in highly reputed journals. What is the most amazing is he is collaborating with several universities all throughout the world. Just to name a few: Hebrew University of Jerusalem, Israel; University of Hawaii, Honolulu, USA; Nagoya University in Japan, and so on. Presently, interestingly, his team is involved in COVID sample identification. So all of you participants, I'm sure, are eagerly waiting for this invited talk of Professor Gulab Khedkar. May I invite Professor Gulab Khedkar to present his talk. Professor Gulab Khedkar. Yes, sir. You may start, sir, the talk.
Can you hear me?
Yes, sir. Yes, we can hear. Yes, sir, you may start.
आवाज येतोय का मॅडम माझ्या तिकडे? नाही फोन म्हणजे yes. I hope you can hear me perfectly, and there may not be a problem because I can sense from here my voice is audible to you or not.
Yes, sir.
If some
We can hear you.
problem persists, you can directly call me on my phone number so I can just get in touch with you.
Yes, sir. We can hear you, sir.
With a hope that everything is all right, I will begin with my lecture. First of all, I take opportunity to thank the organizers for organizing this nice initiative and this the series of lectures. I thank Professor Bhavita also for inviting me and participating me in this initiative. I am sharing with you a very small story and that related some of the deliberations we did during past 10 years. Small stories, big impacts, DNA in life science research. The situations are so crazy all across the world, and our whole life, everyone's life is now just revolving around almost 68 wars. And this is becoming like a, like day to day, every moment we are coming across with these, words which were never been concerned to us. And they become like a part of our life. So with the hope that everyone is comfortable and have this, crazy situation be set over very soon. In that regard, I will have few things share with you regarding the COVID-19. And then I will, after these 4 or 5 slides, then I will switch over to my lecture. Everywhere we are talking about like a bad thing about whatever pathogen we are having now, this virus, COVID-19 or corona. But trust me, pathogens are not always to kill us. They are also essential part of our planet and they are essential part of our life. So no wonder pathogens are really not there to kill us. But if you look at the situation between human or host and pathogen relationship, from the beginning of life on this planet, pathogens and other hosts, they always go hand in hand. That means we are matching the evolutionary rate with a similar space, and that's why both are existed till date, hosts as well as pathogens, and both require each other. And this is a natural phenomena. And whenever such phenomena is going, it is called a natural, and everything is perfect. Sometime what happened with the advancement of research in life sciences or medical sciences, somewhere we try to elevate ourselves much better than the pathogens or the whole, whatever the parasites we are having around us. We are talking about pathogens now. We'll talk more precisely about the COVID. So in this situation, what happened due to the advancement of many drugs, then therapies, somewhere we try to surpass the pathogens, and although it is a natural phenomena, but artificially we elevated ourselves in combating the pathogens. And this is also okay, although it is artificial, but no worries. This phenomenon is perfectly, going. And this was like, we are feeling so happy with the advancement in the medical sciences. We are feeling so happy. We are longevity had been increased, and human being can, combat any disease situations. But all of a sudden, during December 2019, what happened? The pathogen which appeared or which, emerged some part of the world, which is having something very different than natural phenomena than what we are talking about, the hand in hand or the evolutionary rate with host pathogens. Somewhere it is mismatched. And this happened first time in the history of humanity. That pathogen which came, which is having faster evolutionary rate, and which is having like something which is not going naturally, and it surpassed human beings. And result is enormous human, toll of human lives, as well as enormous impact on the human lives, everyone's life, everyone who, no matters which country you are living, life is really changed. And this appears to be not a natural phenomenon. This naturally will never happen. Some pathogen may suddenly appear and which is having very, different evolutionary rate, which you don't have, your body don't have answer for that. It is serious. And we must be careful in these situations. So we must be careful in a sense, because in my opinion, we can't see any hope in, quick time or quick space of time. We can't see any hope. We may have some vaccine against this pathogen, because it is evolving so rapidly. It is evolving across the populations, across the communities, across the country. Evolutionary rates are different, and their genomes also, correlating genome becomes so difficult. And that's why only one thing in our hand is to take care, keep ourselves safe, and have safe life. With this, wish, I switch over to my today's lecture. Before that, I must share with you one thing. Like whatever we are talking about the pathogens, we must be positive about it, because without pathogens, whatever our immunity may not have elevated to this extent. Whatever immunity we are having, it is a gift of, some of the pathogens. And that's why on positive side, we must think that one day we'll have better immunity than what COVID we are talking, what coronavirus we are talking. We may have better immunity in coming days. And if it happens, we may have, we will get rid of so many other pathogens in elevating our immunity. With this thing, I will switch over to my lecture. The small story, big impacts. So in this small story, big impacts, I'm sharing with you, here power gone in the room. So So small story, big impacts. I will share a very classical example of one, very small initiative we started during 2004, 2003, 2004. And it's a revolution driven by a short stretch of a DNA, or short stretch of a mitochondrial DNA. Just 650 base pair DNA segment we used for, or we called it as a DNA barcoding. It is having enormous impact on the, several field of life. And this initiative begin with very few people working among in that initiative. And then successively we form an international barcode of life consortium. And in a very short period of time, like a 10 years time, almost 600,000 species we could able to name. If you look at the natural speed of identifying species and giving name to them, it was 1.7 million species we could able to identify and recognize them, which took almost 250 years. And contrast to that, within 10 years, using this small segment DNA barcode technology, we could able to recognize 600,000 species. It means we have reduced the rate of discovery of species to almost 200-fold. And this 200-fold difference was very much essential, because a lot of species we are wiping every day, and species are very important. Take example of corona. we were having some means to identify corona. That's why we were able to recognize it and immediately alert the societies across the globe. But using this DNA barcoding, what we could able to do during just 10 years, this could be one classical example of this in the science or scientific inventories. Take example of space science, or take example of even like a Big Bang theory like things. Whatever publication resulted out of such, such fields of life sciences, DNA barcoding is equally contributed to that. Here we could able to have published almost 12,000 publications in over a period of 10 years. And all publications related to DNA barcoding are having more than 100,000 citations. This is one of the classical examples in the space of science, and which is having a big impact. Although technology is very simple, very small, very crispy, but having enormous impacts. If you look at the publications, how publications growing, important thing, most of the space technologies in past era were just confined to some of the developed countries like US, America, US, UK, and Europe. But DNA barcoding, if you look at the publication, this pie chart on the right hand side, you will see almost every country is contributing, no matters whether it is a low income country or no matters whether it is a high income country, everyone is contributing. And if you look at the image on the left hand side, you can see here how people collaborated, how people communicate each other, how exchange of knowledge happen, how publications happen, how institutes get connected is an amazing thing. Over a period of 10 years, how many collaborations could happen, which is unimaginable, which has been done by a small technology or small fragment of DNA created a big history. And it has hugely advanced our capacity to understand species interactions and their impacts on human activities. This is amazingly happened with a technology which is called DNA barcoding. Very small, very simple technology, having a big global impact, and years to come, this impact will go on growing in an enormous way. So with this basic introduction, I would like to share some of the stories which been created in our institute over a period of last 10 years. So we entered into DNA barcoding and created several scientific stories. Out of that, very few stories I am going to share with you crisply. So first story ever happened in the scenario of India, when whole river system is scanned. After Britishers, this never been happened, mapping the diversity of a species. It never been happened before. Some river system is scanned for its species diversity after Britishers left India. So first time we selected Narmada River Basin, one of the important river systems in India, and we scanned whole river system and found amazing results. The scanning been done using DNA barcoding. We took all the fish species and we DNA barcode them, and amazing results came. Because the earlier studies, whatever they were talking, like one study been done in 84, which was showing like 74 species. Another study was showing like 29 species. And when we compare these 2-3 studies together, our study found 85 species. When you compare them together, we just found 29 species common to that. That means there is a enormous ambiguities happen in the species recording, species mapping, and creating their checklists. So we scanned whole river basin, and first time we could able to set the species boundary for fish from Narmada. And in this species boundary, we could able to resolve the taxonomic ambiguities associated with almost 17 species that were never been, classified according to their real taxonomic positions. Based on genomic data, we could able to do this, and wonderfully, the database or first scrutinized library can be created for Narmada species. Looking at the success of this story being completed, this is a very small segment. One of my students, she did whole this research in 3 years, And looking at the amazing results and amazing output of this story, we switch over our efforts to map whole river systems in India. And in that wake, we completed Godavari river system, we completed Krishna river basin, we completed Mahanadi, Narmada already we did, and then in those Indus river system we started, and now some efforts we are recruiting on doing Brahmaputra as well as Gangetic river systems. And with a hope, whatever efforts we are recruiting here, hope that one day we will be able to dictate the geological history of India using fish as a model, where we are using small segment of DNA. It may be one of the classical example to map diversity of species in a very short period. We're using just one group, one laboratory involved in doing these studies. Only it could able to possible because we are involving a small segment of DNA in correlating this diversity of species. Wonderful research articles came out of these stories, and wonderful new things we could able to tell to the scientific society. Simultaneously, looking at this initial success on the fish discrimination, we started mapping the marine diversity of fish, and this became one of the best institute or one of the best reference library for marine species in India. And we collected East Coast as well as West Coast, and not only that, we have collected the specimens from Andaman and Nicobar Islands, and all species database has been created, which is ready to use and ready to explore further. One day this database will prove to be one of the important database, because whatever species been wiped or whatever species are at the extinction, at the verge of extinction, can be correlated using this story. Not only fish diversity, we could able to do some of the basic scientific insights we bring out about using our deliberations. One of the classical example is, up till now, till 2018, we are thinking DNA is very fragile, and it denatures at a temperature of 100 degree or near to 100 degree. We conducted one study where we could able to authenticate the origin of meat species from all Indian culinary procedures, where food is cooked like roasting or deep frying, temperature is reaching to 172 degree. And even whenever DNA is present in a tissue, and if you are even processing it 172 degree, DNA remains stable. You can see the images on the left hand side, which will show you at 172 degree also we are getting intact DNA, which could able to track the species using DNA barcode. That means DNA barcode is beyond imagination, is successful technique in even tracing the genetic or tracing the biological species at even very high temperature. And it has proved that DNA is highly stable molecule even at high temperatures. So this is one of the nice lead we could able to do. This study is having enormous implications in the food processing industries as well as in consumer satisfaction, and following the food traceability issues associated with the traditional foods in India, as well as processed food which is sold across the supermarkets in global. So this is a second example. Very simple thing we used. We processed every food materials using the professional shapes, and scientifically temperature been measured and try to correlate whether it can happen, whether it is possible to trace, do your kebabs or do your biryani, or maybe like a fish fillets, fried fish, can be trackable using genomic techniques. And with 100% confidence, we demonstrated this is possible. So back of the mind, we are giving a consumer confidence using this technology. Third important story we created about a very important and highly expensive, spice condiment, the saffron, which is one of the most expensive spice across the world. And in India, it is produced and exported. Like 70% of the saffron is produced and exported to the whole world. when we try to see whether really it is a original saffron or it is a spurious, results are very shocking. We found in Indian markets, whatever saffron we are getting, you will never get quality 1 and quality 2 standard saffron, you never get. In Indian market so far, whatever 86 samples we collected, we found no single sample can be correlated to the first or second grade standard as per FDA. And we found only the substandard saffron present in Indian markets. In that also, almost 49% saffron is totally spurious, nothing to do with saffron. And a lot of adulteration happening across the country. So whenever someone is buying saffron, he can look at this article and avoid and usually like protect yourself in getting to the market for buying the saffron. Even we developed 2-3 nice insights about this. One simple, even your mobile based detection of original saffron, you can see the images on right. And another thing, we correlated originality and quality standard in genetic terminology. So looked at the genetic purity, you can correlate the originality of saffron. So this nice insight we could able to do, and has opened a new topic for the researchers working in the area of food processing. They can test such type of many products available in the market which we are using as a spice for various processed products, and this can help human beings. This can help the common person. This can help consumers in protecting their rights. Similarly, another story we could able to create using this small segment of DNA, or called DNA barcoding, we could able to reach very perfectly. We could able to address the wildlife forensics, and we are providing nice services to wildlife forensic services to the wildlife department or the forest department in Maharashtra, where several cases we handled, and so far out of 28 cases we handled, almost 9 convictions could able to happen. And this is in the wildlife forensic, this is the first example where out of 28 forensic cases, 9 conviction happens, and still half cases are pending in the court of law. So earlier, the conviction rate was very less. And using the small segment DNA based technology, which is pretty convincing to the court of law, and has able to prove beyond doubt the cases registered against the culprits. So few examples just I'm showing here. Like one example was associated with the claw, which been sold in the market nearby a city in Nashik. Another one was a skin, and that nicely we could able to bring these stories to the notice of the world. So coming from your ecological systems or the river systems, we turn to the food. From there we could able to reach to the oceans, and from there we could able to reach even the forensic science or wildlife protection. And now one component of wildlife protection is a snake. So relatively, the research in the field of snakes was very scanty in our country. A lot of people, since snakes are protected under the Wildlife Protection Act, and it is very difficult also to handle them and collect them from their natural habitat, it was becoming challenging. And that's why we were so primitive in case of snake research in most of the countries, not only India. We demonstrated that the skin exudate, you can see the first image here. So skin exudate can be wonderfully used in population genetic studies, in identifying species, in locating the habitats of a particular species of fish, particular species of snake. And this story has proved very nice. And based on just using this skin exudate, we started the first gene bank for the snakes in India. And this become this is a part of community science. We ask people to deposit wherever they will get such type of skin exudates. So there is no ban on handling skin exudates as per the Wildlife Protection Act. So you can easily post them, you can handle them, you can preserve them, you can put in your laboratory, or you can extract DNA and do your studies. There is no obligation from the Wildlife Protection Act. So one powerful tool we could able to put in the hand of researchers who are interested to work with the reptiles and specifically the snakes. Then very important story I must share with you by touching DNA in our research. Very first story we started in 2006-07. That time I just did a RAPD based analysis of Clarius batrachus, one of the important species for aquaculture. And we were thinking, because this fish was that time, like 2008-10, from like 2006 to 10, fish were becoming scanty and a lot of fishermen were, you know, like talking about the fish. It has prompted us to see whether this fish is having something to do with its DNA because they are declining. And for that purpose, we did simple techniques and we found the genetic diversity lacking with this fish across the country. And then, you know, we started looking into this fish, why genetic diversity is not getting. And we came across like 3-4 stories and sharing 3 of them. So next we planned really why it is happening, why genetic diversity is going down. First method we used was just RAPD, which was so primitive. So we want to see really what we are correlating is correct or wrong. And that's why we are, you know, develop one proposal to Department of Biotechnology, and we got funding for, you know, detecting the genetic diversity of Clarius batrachus in the country. And for our surprise, within 4 years, the fish become almost unavailable. And we found, you can see these images here in the middle, Clarius batrachus is almost now substituted by the another species which is called Clarius gyripemus, which is very much similar looking. And you know, one cannot figure out whether Clarius batrachus, what you are looking, is similar or it is a different species. There are many taxonomic characters described were found to be not applicable in case of Clarius batrachus species detection. And this species has completely now, replaced, almost completely replaced by the exotic species called Clarius gyripemus. Then again, we were in the same study, we were interested why these things happen so rapidly. And then we did like a microsatellite recently, 2020, we published this article where we genotype the populations of Clarius batrachus, whatever available across the country. And one surprising thing came out, genetic diversity loss, and many domains we could able to see genetic diversity. One is a whole nuclear genome, another is a mitochondrial genome, and third time we did based on their microsatellite based studies for scanning whole genome, we found genetic diversity has gone down like anything. And this is one of the important reasons this species is wiping. That means in very, very short period, I may not be wrong, maybe within 2 years span of time, you will not get any Clarius batrachus in our country. And why it is happening, when we try to reach at the base of the issue, we found in Indian scenario, we are having seed regulation policy, but we don't have seed regulation policy for aquaculture species. And there is no policy. That's why what is happening, there were 2 main places which are producing seed for Clarius batrachus. One is Kakinada and another is Kolkata. And all this seed is coming from these 2 places and spreading whole country. That's why these fish reaching to the natural habitats and they destroyed the natural genetic diversity of stock available, wild stock available in the natural resources. And that's why how important is reforms in aquaculture, how important is reforms in the hatchery technology that has been underlined by this study. And you can see both the images which are pretty convincing. This fish may not survive now for more time. And this is one of the example government has to rethink on stringent policies in deciding the aquaculture practices and in deciding and in regulating the hatchery practices. Whether one hatchery can supply seed to whole country, this should never happen. Otherwise, the story happened with Clarius batrachus with of aquaculture. And if it happens, so one day we will lose almost all aquaculturally important species in our country. and we may need to import some species. We may need to bring some species in, like what happened in the case of Penaeus monodon. Almost we are stopping the culture of Penaeus monodon, and now we are importing the Lithopenaeus vanami from Hawaiian Islands because it is pathogen free. Whatever pathogen, whatever the tiger shrimp we are having, it is having a lot of virus pathogens with that, and that's why we have stopped aquaculture. India was among the top second ranked country in shrimp production. Whole industry collapsed during the 2000 that decade, and now we are revamping it. How much wages, how much man days wasted, how much foreign currency, and how many bank debts, and there are so many ugly things happen with the Clarius, with the Penaeus monodon. Same story may happen with other species of these aquaculture important organisms, and for that purpose, this study become a role model. This study become one of the important indicator in deciding the policies. So, being a life science research, not only we can do research for getting the publications or getting our promotions and getting jobs, but we can even participate into the national policies. We can give important insights to the policymakers, and we can even protect our diversity by developing nice resources and literating the policymakers. So DNA-like molecules are really important in such initiatives. Then along with, not only we stop here with the looking at the genetic diversity, but we are going further. What might have happened? What is the implication of lowering the genetic diversity in Clarius batrachus? And we try to scan out certain type of important housekeeping genes, and it was surprising. The housekeeping gene, which is called to be highly stable housekeeping gene, and they don't have much variations, but in Clarius species, we found 3 type of housekeeping gene and their promoters having highly diverse architecture. Usually in housekeeping genes, it never happens, but this story happened in case of Clarius batrachus. Every population is independently evolving, and even their housekeeping genes and their promoters also are evolving so rapidly. This manuscript is available on BioRix. One can look in that and one can comment on that. So this another insight with a little bit higher genetic considerations we could able to create. So identifying species, looking at the substitutions, looking at population genetics, followed by looking into the different genomic sections, looking into the policies, and now we reached into classical genetics associated to the species which are under threat. So small molecule can tell many stories, which is perfectly applicable in case of Clarius gariepinus, what we did for last 10 years. Similarly, not only doing this, hello? So by I was thinking the voice may not be clear, that's why I took this phone. So similarly, not only we are stopping here, but we are keeping our eyes open and ears open to see what is happening around. And one day we got an attention because in one newspaper they mentioned like piranha, red-bellied piranha is entered into Indian waters, and that too very close to our place, Godavari river system. And I was so much shocked to looking at this news, because if red-bellied piranha is entering into Indian waters, it may create havoc. And then what happened, I tried to look into many aspects, many newspapers. We scanned out available literature, and we found there were almost 15 news published which are mentioning across the different newspapers in the country, print media, and they mentioning the red-bellied piranha is entered into Godavari river system, Mahanadi river system, then Krishna river system, Narmada river system, even Gangetic river system. And if in this speed, if piranha is entering, it may be a great danger to the aquatic diversity as well as the people who are living, who are having their livelihood on these resources. And I felt this is very important to get involved. And we collected the samples of red-bellied piranha. It was so surprising. When we completed this study, we found so interesting results that red-bellied piranha, what media is talking, is actually not red-bellied piranha, but it is a subspecies of, not subspecies, it's a different species of piranha, which is totally herbivorous and which is called pacu. So red-bellied piranha was not available, and whatever print media was publishing the news, it was misleading to the community. And that's why this paper became one of the classical paper mentioning that red-bellied piranha is not present, and media should be careful in reporting some of the things which are related to science and which are related to the basic science. But while doing this study, we found one important understanding, one important insight also. Not only in India it happens, but even it happens in Brazil, it happens in Africa, it happens in other South Asian countries. Piranha means it has identified with different names, and there are two subpopulations associated with red-bellied piranha in Brazil also. And just by mining the information about the genomes and contacting few scientists, we could conclude even the red-bellied piranha is having two different clades. They are like two different species they are having, although that are not present in India, but red-bellied piranha may be classified into two different species. And you can see the bottom here. So these are the two different species of red-bellied piranha, 8 not being segregated, even the people who are living in vicinity of the Amazon River. That ambiguity of the species diversity or species identification we could able to create sitting in Aurangabad in India. Why could we could able to do this? Because we are getting the access to the information, very short information called DNA barcoding through the genomic resources. How DNA is powerful, this is a best example how DNA technologies or how implication of DNA in life sciences is powerful. This is a best example. Then I'm just reaching to the last section of my lecture. I will just share 2 stories and then I will conclude my lecture. So recently we switch over, not only we are looking at the 650 base pairs, but we want to increase little more horizon and to see that what redundancy can we receive by studying a bigger region of mitochondria or whole mitochondria and small section of mitochondria. And so nice information we could able to get. First whole genome we did for Hanuman langur and Simnopithecus hypolecus. This is one of the endangered species in the India. It is this is confined to the peninsular Western Ghat region. And by doing this whole genome sequencing, one nice thing we could able to do, because getting samples from Hanuman langur, again it is a protected species and you need to chase them in the forest is impossible. You just track them on their way, and we just collected few of the fallen hair follicles. And from that, we could able to amplify whole mitochondrial genome without, requiring any ethical considerations, without requiring any, like a Wildlife Protection Act regulations. We could able to do this non-invasively. And this small information has created a big story. And we found in case of primates, cytochrome oxidase 1 is the wonderful gene, best gene in clarifying the taxonomy of the species. And by doing this, we could able to set the species boundary at single genome level, single gene level, at different genes, at concatenated protein coding, 13 protein coding genes, as well as whole mitochondrial genome. And we found the 3% boundary for species recognition is perfect. Either you use single gene or use a whole mitochondrial genome. This insight has been first time developed, and we could able to solve the ambiguity associated with the Simnopithecus for over 2 centuries. And now we proposed Simnopithecus and Trachopithecus are not 2 different species, but these are 2 different, these are single genus Simnopithecus. And misidentification happens with Simnopithecus joini is Trachopithecus joini is clarified in this study, and we mentioned this is not a Trachopithecus, but it is a Simnopithecus joini. So in this way, Hanuman langur clade has been cleared. There are 6 subspecies associated with Hanuman langur, and people were suspecting Simnopathicus hypolochus is also a subspecies. But by doing this mitochondrial genomics, we could able to tell this is not a different species, but this is the similar species, and it is a complete biological species, not a subspecies. So that was nice insight we could able to bring out using just doing a small hair follicle sample and doing a whole mitochondrial genome. Similarly, we touch one of the most important species found in India. And these are like freshwater dolphin. Only 3 species of freshwater dolphin you will find across the country. All these 3 dolphins are restricted to three different continents. One of them is a Platanista gangetica, which is found in the Gangetic River system, which is popularly known as Gangetic dolphin. So in Gangetic dolphin, we could able to collect the specimens from the forest department, and by doing genomic analysis, we found whatever people were talking like marine dolphins has entangled into the freshwater and they become freshwater dolphin over a period of time, they evolved and become freshwater. But when we correlated the evolutionary histories associated with different mitochondrial genomes, you can see in this figure on right hand side, the river dolphins and marine dolphins, they evolved at different rates. Their mitochondrial genomes evolved at different rates. Their evolutionary, rate of evolution is very different. And it shows we need to reconsider on thinking whether it is there is something to do with marine dolphins, freshwater dolphins and marine dolphins. Do they have any correlation? We need to reconsider it. This study is clearly showing there is very faint link. Even you can say there is a link between these 2 species. Then another interesting thing was coming, like almost over 250, almost 300 years, people were considering Gangetic dolphin. There was a lot of ambiguity, although this is a charismatic species and having a lot of importance mythologically, then ecologically, and even globally, this was having enormous importance, but we have never paid much attention on this species. And this is under the great threat of extinction. So this was one of the purpose we started doing study on this. So what we found when we correlated the genomics of this species, we found there was a lot of ambiguity. Still now people were not clarified this taxonomic status of this species. And to clarify the taxonomic status of this species, it was very important to have some sound background of, about the species, because the species which India are having, there are 2 species. One is Gangetic and another is Indus River. So some scientists believe this Platanista gangetica is one species. Some believe these are 2 subspecies. And they name Gangetic dolphin as Platanista gangetica and Indus as Platanista gangetica minor. When we gathered genomic information and analyzed them, we found very interesting thing. Absolutely, there is no chance doing mitochondrial genomics. There is no signal to differentiate these 2 species. They are not differing at all at the genomic level. Then question is coming, how biological species can be defined? As per the theory of Darwin or Linnaeus, we need to breed them, and then if they are giving the viable offsprings, then you can correlate this is a species. So this is almost appears to be impossible to bring these 2 species, male, female, and breed them and then watch them, because they mature very late, like 10 years. So it becomes so difficult. How to find out whether there is a reproductive isolation or not? Our study has bring one nice insight. Genetically, these species are not different. Not only this, by reviewing the literature, we found some clues that the Brahmaputra, Gangetic dolphin and Indus dolphin appears to be one basin almost like a million years ago, 1.3 million years ago. And it gives one signal that this species, which is present in the headwaters, which is originating in the Brahmaputra, is flowing through Gangetic basin and reaching to the Indus River system. And whenever there was a continental shift happen, and due to continental drifting, theories, the basin has been separated. And that's why this one population of Platanista gangeta is divided into 3 populations. Now question is coming, in Brahmaputra you are not talking as a subspecies, then why you are talking the Indus dolphin as a subspecies? Brahmaputra and Gangetic dolphin, we are not giving them subspecies status. Then why you are giving substatus to the Indus dolphin? This was a question, and by correlating this whole study, we proposed that subspecies hypothesis should be abandoned in case of Gangetic dolphin, and both the species should be treated as a single species, Platanista gangetica gangetica. Just based on their geographical isolation, one should not call this as a subspecies. There are many papers which are criticizing the subspecies status. So this is a role of scientists to need to think on classical, and sound background, whether old concepts should be continued or they should be criticized or they should be rectified. So I'm happy to share, we are proposing to rectify a very old concept, like almost 100-year-old concept, where Platanista gangetica is designed as a subspecies and two populations were described. Just because they are found in two different river systems. One belong to Pakistan and another belong to India. May not be, because the time frame which has not allowed these species to evolve, and in my opinion, and based on this understanding, these evidences, I'm sure if you breed Platanista gangetica minor from Indus River and Platanista gangetica from Gangetic River system, I am 100% sure they will give viable offsprings. This is one of the hope we can regenerate the Gangetic River population, which has declined drastically. And I think there may not be better contribution of science than this, when we can bring some species to the, flourishing its populations. So with this hope, I am sure, and everyone also, we know the importance of DNA barcoding, not DNA barcoding, the DNA research, which is having enormously implications in addressing the basic and applied questions in life sciences. So my friends, the young researchers, PhD students, master students, graduate students, try to correlate your research, something related to DNA, which can tell you so many stories. Which can bring you the ancient past in correlation to the present scenario, which can answer so many questions which have been not answered by even scientific giants. You can answer them. This is a need of day. We need to dominate the science. Biological scientists are doing miraculous work. Humanity, you must be knowing now in COVID situation, everyone is looking at the development of a vaccine. Here, space scientists are not contributing. Here, like your other branches are not contributing. Here, only biologists are contributing, medical scientists are contributing. We are, being a biologist, we are great people. We need to involve some of the, universal molecules in our research in addressing the questions. I thank all of you for listening me, and I'm thankful to the organizers for giving me opportunity to share my views with you. Thank you very much.
Thank you, sir. Thank you, sir, for your enlightening talk. And your lecture spanned through all the taxa. They, in fact, even plant. And I have some questions. We've seen some questions in the chat box. There is Viba Gupta. Her question is, please let us know the app used for identification of pure saffron. This means we are using substandard saffron.
Any questions or anything, please share with me. But I can't hear any voice from anyone. I can't hear you. Amol.
Hello. Sir, Bhavita madam is talking.
Amol.
Hello, sir. Hello. Khedkar sir, hello.
Amol, can you hear me?
Yes, sir. Yes, sir. I can hear you. Hello.
I'm not getting any voice from your part.
Okay. Okay. Hmm. Hello. We can hear you, sir. Hello. Hmm. Hello. Hello. Bhavita madam is also not in the meeting now. She has left the meeting. Hello. Hello.
Hello, Dr. Khedkar. There was a question from the audience.
Hello, sir. Khedkar sir is not getting our voice. Hello.
Khedkar sir. Hello. Dr. Khedkar, I'm audible to you. Okay, I think there are some technical issues with the connection.
Yes, sir.
Hi, yes, sir. So this is Dr. Saraf, and I think due to some reason, Bhavita madam could not join the group. We have certain questions from some certain queries from the participants, and there was a question regarding the identification of saffron.
Amol, I can't hear you.
Sir, can you hear me? Dr. Khedkar, am I audible to you?
Hello.
Hello, Dr. Khedkar, am I audible to you? I think, dear participants, we will post your questions to Dr. Khedkar, and he'll surely reply back on them. So I think there are certain technical issues. So we shall move to our next speaker. Dr. Hello, am I audible to you? Amol sir, am I audible? Amol sir, am I audible?
Hello, sir. You are audible. Hello.
Okay. Okay. Thank you. So let me take this opportunity to thank Dr. Khedkar for his very informative speech. Sir, we have many questions lined up from our delegates, and we'll be emailing you all those questions, and I'm sure you will be replying them back. So thank you, Dr. Khedkar, once again. There are some certain technical issues. So Dr. Bhavita could not join us back. Thank you very much, sir. So now we'll move on to our next speaker. Good afternoon to all. I would like to welcome you for the second talk in this session of the conference. Dear delegates, I would like to welcome our next speaker for the session, Dr. Jacinta Dsouza. To tell you about her, after doing her graduation in microbiology from Khalsa College, she did her PG and PhD from TIFR. from 2007, she started teaching as an adjunct faculty in UMDA Center for Excellence in Basic Sciences, Kalina campus. And she liked the teaching so much that she actually took voluntary retirement from the TIFR and joined as a reader in the DBT ICT Center of Energy Biosciences at ICT Matunga. Then she continued as a reader in again UMDA Center CBS from April 2010. After becoming professor in 2017, she is now a chairperson of CBS since 2018. Dr. Jacinta has a very commendable academic and professional to her credit. She has been invited for more than 50 plus talks. She has organized 4 workshops and 35 educational trips. She is recipient and she has memberships from various scientific societies. She has received nearly 22 research awards for her presentations. She has more than 30 papers to her credit in reputed national and international journals. Currently, she has 3 students registered working under her for a PhD, and 5 students have already been awarded PhD under her guidance. She has also guided 40 MSc students for their dissertation, and she is also part of 12 UMDA CBS report. So with this brief welcome, I think that is sufficient to give you the kind of experience our resource person has. So with this short intro, I invite Dr. Jacinta to conduct the session. Dr. Jacinta, please.
Thank you, Dr. Saraf. Thank you very much. I'll start sharing my screen. Please let me know whether it is visible to you. Is my screen visible to you? Dr. Saraf? Am I audible? Dr. Saraf?
Yes, madam, you are audible. Hello.
Okay, okay. Is my screen being shared? I mean, is it?
Yeah.
Okay, okay. Thank you very much. So at the outset, I'm really very glad, and I thank the organizers for considering me as an invitee for this very interesting international e-conference. I'm glad, and thank you, Dr. Saraf. I'd like to shed some light on the tool of CRISPR-Cas9, which is now a very popular genome editing. It's no more new, I must say, but I'm very tempted to call it new because many people are have adopted it, and many people are using it for various genome editing purposes with the organisms that they are investigating. So, without much ado, I'd like to tell you what would be the flow of my talk. Since there is a large population of students in the audience, I am taking the liberty to introduce a few molecular biology basics. And then, something that is very close to my heart, that is a scientific investigation of my lab. I'll use my scientific investigation as examples to understand genetic manipulations. And because CRISPR-Cas9 is a reverse genetics tool, I'll start with some reverse genetics tools, some examples, and then state the drawbacks. Ultimately, I will give an explanation of the history, the biology, and the technology of CRISPR-Cas9. So let's go to the basics. We know that molecular biology is a vast subject now. It is a study of molecules that are present inside cells. It's a huge branch of biology that studies gene structure and functions basically at the molecular level. And because it needs to study several molecules inside the cells, inside a cell that is crowded with a lot of organelles and many other substances, it is a field that overlaps with several other areas such as cell biology, genetics, and biochemistry, to name a few. And of recent, bioinformatic analysis has helped us immensely in predicting the nature of some of these molecules, particularly DNA and RNA, and what kind of structures proteins can take. That takes me to the most popular molecular biology dogma, wherein DNA, which is a master molecule, can replicate itself by a process known as DNA replication. DNA is also used as a template in the process of transcription to form RNA molecules. The messenger RNA is the molecule that is used in the process of cellular translation to make a myriad of proteins, a heterogeneous lot of proteins that take various shapes and sizes inside the cell. This heterogeneity also gives all these proteins its dedicated functions. And therefore, structure function is something that one must understand very well to know the encoding, to know the function of the gene that is encoded. And therefore, several molecular biologists have been studying major topics and some minor ones, but I've listed the major ones here, such as recombination, a process which occurs during meiosis and wherein there is strand exchange. Such recombination, such DNA recombination has immensely helped develop several tools and techniques which have also been used by molecular biologists to address cellular processes. Followed by gene expression and regulation, primarily where the DNA, as I mentioned in the molecular biology dogma, is the template for transcriptional control, wherein the primary transcript is made, which is further processed by RNA processing control. The mRNA finally makes the protein. At each point, there is regulation of these steps. And this is also a main topic that several molecular biologists have been investigating. Further down, when these proteins are made, each of these proteins, as I mentioned, have their own functions. They have their own structures. It's not just proteins, but several fatty acids, carbohydrates, as well as nucleobases have their dedicated functions inside the cell. Several biochemical and biophysical techniques overlap in order to understand the structure and function of these macromolecules. And therefore, this gives rise to the systems biology approach that many molecular biologists have now adopted to study in totality the genome in a field known as genomics, the proteome in a field known as proteomics, and so on and so forth. Therefore, this leads us to the understanding of several cellular processes that occur inside the cell. And for dissecting these cellular processes at the molecular level, one needs to look at the functional analysis of these genes. One needs to isolate these genes, make the protein, study the protein maybe in isolation, and then make variants of these genes so that you understand the function of these genes and proteins better. In a nutshell, functional analysis of a gene is something that has helped us understand cellular processes in detail. And therefore, what we say is every gene matters. The cellular process that we are interested in our lab, which is situated on the Kalina campus of CBS, of University of Mumbai, is motility. We are trying to understand how cells move. Most of you might know that cells harbor very tiny projections on their extracellular parts. What is depicted here are the sperm cells, which have these tiny tails. These tiny tails literally push the cell body of the sperm towards the ovum, giving it what is known as the lashing type of movement. This is one type of motility. The other type of motility that is seen in several lower organisms, such as the protozoans and the paramecium depicted here as an example, have several flagella or cilia around the periphery of the cell. And this gives it a coordinated movement such that such a paramecium or a protozoan which harbors these cilia can move towards its food or away from any meat. More closer to humans, we have the human airway epithelial cilia present in the respiratory tract of the human body. If you can see these tiny hair-like structures that are present on these epithelial cells are those ciliary structures that I'm precisely hinting at. These cilia are all therefore present on the sperm, the protozoans, as well as the epithelial cilia mentioned here. These cilia help in their brushing movement, they help move away toxins, dirt, even viral particles and bacteria outside our system. And therefore, such cilia become a very important point of investigation, a very important program for scientific investigation. So where are all these present in the human body? We refer to these types of cilia in the classical terms or the typical terms as the 9+2 that is mentioned here, and the details I'll share in a while. These 9+2 are present in the ependymal brain cells of the human body, in the respiratory tract as shown here, and also in the reproductive system of human, the male and the female. So we have been using Chlamydomonas reinhardti as one of the systems for investigations. We have been long using this particular organism as the primary model organism of investigation. Because it has these two flagella, if you can see these fine thread-like structures that are jutting out of the cell, known as flagella, a tiny eye spot which detects light. It being an algal system, has a huge chloroplast, a cup-shaped chloroplast, and obviously a nucleus because it's a eukaryote. For long, I must say, from the 1970s onwards, the flagella of the chlorophyte, that's Chlamydomonas reinhardti, has been investigated and has served as a very good model organelle for understanding ciliary diseases that are affecting human beings. And therefore, sometimes in 2007, the genome of this very tiny algal system, unicellular algal system, was sequenced, annotated, and over the years, around 70 to 80 years, several flagella mutants have been isolated by the very techniques, that I mentioned as reverse genetics, by the tools of reverse genetics, some of which I will show you in subsequent slides. This particular organism is very handy. Although I cannot see the cartoon on my presentation, I hope some of you can see the cartoon. Chlamydomonas moves in what is known as the typical swimmer's breaststroke movement. And the homology between Chlamydomonas and the human proteome is to the tune of 4,000. I must also say that this flagella of Chlamydomonas and of the human share a very high similarity of proteins between them. Not only the structure and the architecture, but also there is a high conservation between the protein sequences. So what are we studying? What are we trying to understand? So here I have shown you a vertical and a transverse section of the so-called 9+2 flagellum, the extracellular organelle which I showed you in the previous slide. Let us see what it looks like. I'm going to spend a fair amount of time on this slide. I would like to draw your attention to this organelle. It is typically called as the 9+2 organelle because of the 9 doublets of A and B filaments that are present. If you can see those filaments over here, they come from the basal body, which is anchored to the cell and juts out. So these are the filaments that I'm referring to. And this is the cross section that is presented out here. These doublets are nothing but microtubules with one complete doublet, with one complete microtubule known as the A tubule and an incomplete microtubule known as the B tubule. So you have 9 such pairs in the periphery of this particular organelle. Jutting out of these doublets is this green lollipop-like structure known as a radial spoke. And since it juts out from these 9 structures, you basically have 9 of these radial spokes. These radial spokes basically have a head which terminate towards what are known as the projections of the central pair. What is the central pair? The central pair is also a pair of singlet microtubule. Like the complete microtubule of the doublet, this is also a complete microtubule. And there is a pair, and therefore it is referred to as a central pair. It is connected by protein bridges, shown here in 3 black hyphenated structures. Now let's come to these projections which are marked in yellow out here. These projections jut out of this central pair. And this is the this is the structure that we are interested in. Before I come to the details of the structure and what we are trying to understand, let me also tell you that this is like a micro machine or a nano machine. And since it is a machine, it has to be powered. It has to get some fuel for its activity, like a car, which needs petrol or diesel for it to work. This particular organelle, therefore, moves. That's the mechanical movement we see because the fuel, that is ATP, is utilized by these red-like horn structures that are shown over here, which are known as dyneins, a mega protein multi-subunit complex. They are the ones that utilize the ATP. So the chemical form of ATP is converted into the mechanical form of movement. And there are several hypotheses which actually try to understand the molecular mechanism of motility. What are we trying to do? So we are trying to understand the role of the central pair towards motility. And why central pair? One of the hypotheses states that the presence of central pair is very vital towards the movement of these 9+2 structures. Why do I say that? Because several central pair mutants are motionless, have no motility. And so people are trying to understand the central pair in detail. While most of the other structures have been well understood, the central pair structure and composition is still elusive. So here is the blow up of the central pair and its various projections. We were trying to understand the central pair in such a manner such that we can identify what is known as a signaling complex or a scaffold. This signaling complex is normally anchored to scaffold, such as the one that we identified, known as the A kinase anchoring protein. So I'm not going to get into the detail of the signaling because that's not really the point of that's not really the talk, that I'm supposed to be giving. But what we did, we took an indirect approach. We identified a yet smaller protein. I'm sorry, I cannot see this part of the slide, but there is something written as 174, that is FAP174, flagellar associated protein 174. So we identified that protein by using, again, functional analysis of the gene. We cloned it, we overexpressed the protein, we made antibodies, we used the antibodies to then isolate one particular multi-protein complex, which we found to be situated in the central pair. We found out that there are 9 members to this huge multi-protein complex where 174 is situated. And it spans the C1 and the C2 microtubule. So I'd like you to just remember FAP174 and some of the proteins that are associated with FAP174. So so now we want to characterize. We want to find the role of each of these proteins towards motility. How does FAP174 contribute to motility? how do its partners contribute to motility? So what do? We utilize the tools that are available in reverse genetics. So we are trying to use flagella mutants that are abrogated, that are aberrant in each of these proteins, and therefore mutated in their genes. So in a nutshell, we are using flagella mutants to functionally dissect this complex so that we can understand better the role of these proteins in motility. And that is what many people do. They rely on mutants. So what is a mutant? Again, as I'm confessing that, I'm going to the basics so that students grasp the talk better. A gene mutation is basically a permanent alteration in the DNA sequence. And it is in that DNA sequence which I desire. Like, for example, I desire a mutation in FAP174. And therefore, I make a mutation in the gene that encodes FAP174. Now, you can look at gene mutations in two ways. One, which you inherit. We inherit from our parents, and they are present in the germline. And therefore, they are known as simply hereditary mutations. Then there are the others which we acquire during our lifetime. There are those that even we, can expose cells to mutagens, and, you can acquire these mutations. Most of these acquired mutations are because of environmental factors. Whether they're hereditary or acquired, these are produced in the laboratory either by forward or reverse genetics. And I'm going to delve on reverse genetics because that will lead us to CRISPR-Cas9. So what is the reverse geneticist approach? The reverse geneticist approach is specifically to have a gene in mind. My favorite protein, my favorite gene. You modify that gene. You either modify the gene in the DNA or modify its expression by modifying its promoter or its transcription factor, modify its transcription factor so that its expression is modified. And therefore, study such a cell with its modified gene, with its modified expression, and characterize the phenotype and the consequences of such a modification. In this manner, several mutants have been generated. But there was an era, in the late 1990s, you would have all known as biologists, a large number of genomes were sequenced. And then we were faced with a large number of genes and their proteins whose functions were unknown. And we couldn't predict much. So what do? If I want to predict its function, I want to know its function, I take that gene. I know the sequence because it was sequenced. So I take the sequence, I can clone the gene, I can use convergent approaches to assign a function to those genes. What are those convergent approaches? Identify the subcellular localization of the gene product, that is a protein. Identify the molecular complex the way we have identified, and we know that the molecular complex to which that protein belongs is now in the central pair. Do something like this. And now with those modified or variants, which I'll come to later, I'll explain variant a little later, we describe the phenotype. Because now we have manipulated the gene. And that's how you go about assigning a particular function to your gene and therefore your protein. High throughput reverse genetics approaches are therefore the tools, in predicting the function of a protein. What are those types? So now you would have probably grasped that this is an incredibly powerful tool. What do? As I said, you take your favorite protein, you clone the corresponding gene. Now, I would like you to remember this very well because this is completely associated with CRISPR-Cas9. Take your favorite protein, clone the corresponding gene. These are some steps that one needs to go. Randomly introduce mutations in the gene, one by one. Okay, so one mutation, say substituting an aspartate with an alanine, removing a threonine altogether, creating a deletion, creating an insertion, and you collect a library, make a huge library. How do you create a library? How do you make such variations in your gene? These are the approaches. These are the approaches of creating of making, such variants, such libraries. Each of these techniques can be used. And I'm not going to go through each of them. I'll go through a couple of them for you to understand it a little better. Now, once you have this library, each of these cells in the library will throw up a particular phenotype, right? Because you've transformed these genes with variations into your strain, into your model organism, where it is going to get expressed or not expressed, or some regulation, something has been meddled up. You have changed something, right? Likewise, you then use a very high throughput screen to pick up these variants from the library. And for this, so there are two things you must learn from here, whichever method you are going to use, and ultimately even CRISPR-Cas9, which we are now moving towards. Two things you have to remember very well out here before you even plunge into these. You should know which is your favorite protein you're trying to target, and you should have an output. You should have an assay which will help you pick up these variants, which will help you recognize, this is a variant as opposed to my wild type. It is not a wild type. So there should be something that should tell you this difference. Now, since Chlamydomonas is my favorite organism, I will try to touch upon taking examples from Chlamydomonas and flagella. As I said, this is really dear to my heart. I'll pick up those examples and I'll go through 3 of these techniques. 2 of these I'll be, a bit elaborate, and 1 of them I'll be a bit brief. So, as I said, Chlamydomonas has been an organism of choice to study flagellar motility and flagellar functioning because of the conservation, because of the conserved proteins across evolution and its similarity with several human ciliary genes. Way back in the 1960s and the 1970s, 2 groups conducted a huge reverse genetics screen. What did they do? They took the wild type of Chlamydomonas, they exposed it to some mutagens, in here particularly NG, N-methyl, N-prime nitro, N-nitrosoguanidine. Once they exposed it, they left it alone for 5 to 8 days till they saw some colonies coming up on the plate. Now, these are prospective mutants. Mind you, I call them prospective because I still do not know where the lesion is. Lesion means the mutant. Where is the mutation in the gene? For that, I need to do what is known as a thorough characterization of each of these colonies. And you can rank your characterization. There can be a quick phenotypic characterization because I'm interested in, so since I'm taking flagellar motility as an example, I can just check each colony, grow them in a liquid medium and check for motility. So if I'm looking for an abrogation in motility, I see cells should not move, and so on and so forth. Then look at the cell body. Is there a distortion in the cell body? Is there a problem in the growth? Has it compromised any other component, such as photostimulation in Chlamydomonas could be one such example. I will then check the flagellar length. And several such mutants were already generated with such phenotypes mentioned here. I will not mention all of them. This is one example from a different lab. We have used these mutants from the long flagella cells, from the long flagella mutants of Chlamydomonas to complete one story. What have we shown? We have shown that Chlamydomonas cells with long flagella have one of their dynein subunits compromised. That's what we showed. And we published this work. We use these mutants from such a screen to compile one short scientific story. And that is the usefulness of this particular technique. And there are many such mutants. We have used a couple of more mutants, but I will not share that story with you here. Recently, 2018 to 19, Professor Martin Jonikas, he performed a huge Chlamydomonas screen in a reverse genetics approach, which is known as insertion mutagenesis. And what he did, he generated several cassettes. What is a cassette? A cassette is a plasmid which has defined sequences. And I know those sequences very well. I know the sequence. I know the sequence of the plasmid. So what he did, he put some very short codes which he knew what they are, right? He introduced them. So you can use molecular biology techniques, molecular biology jingoism to modify your plasmid, to change your plasmid, to do whatever you want with your plasmid. So this is what he did. In a very elegant study of primer extension, digestion, and purification, he created such cassettes. Then he generated the backbone of this cassette by introducing this particular barcode in both the 5' and the 3' manner. And then he prepared the plasmid. So this is my final plasmid. The final plasmid has a code here, another code here. It has terminator sequences because it has a antibiotic selection marker and a very strong promoter. Now, this is what you must remember for insertion mutagenesis. So he took such plasmids. He took to chlamydomonas, he transformed chlamydomonas, and he generated those variants. If you remember in my reverse genetic slide, I mentioned variants. So these are the variants he used. He called them mutants, okay, array. He characterized each of them because now I can do a simple molecular biology technique such as PCR with the help of these codes. And that's what he did. To cut a long story short, he could identify where this particular part of the cassette has gone and inserted into the genome. This is random, okay, this is random. It is called random insertion mutagenesis. But with the help of these barcodes, he can now find out what are the flanking sequences on the genome of chlamydomonas. And then further, this is an exhaustive study. He got a lot of photosynthetic mutants, a lot of flagella mutants, cell division cycle mutants, because this was random, and these are the numbers he produced. It yielded 62,389 mutants on 245 plates with a total of 74,923 insertions that were randomly distributed over the chromosomes. Now, while this is an advantage, this is also a disadvantage. Please remember, in one chlamydomonas cell, one such cassette can produce many insertions because this is random, right? We went to the Jonicas collection, which is now known as the clip collection. We procured one particular mutant. If you remember, FAP174, and this is the, this is what is there on the genome. We found out that this is inserted in a FAP174 gene by using very specific primers. We screened it. We did Western blotting. We found out that the protein is not produced in our mutant. We also found out that this particular cell that harbors flagella is extremely short. We also showed that these FAP174 mutants do not move, thereby helping us assign a particular function to FAP174. And this is how the reverse genetics approach has helped us in our research program. Okay, so FAP174 mutants, I repeat, is a null mutant using Jonicas approach. Jonicas is insertion mutagenesis approach, short flagella, and immotile. Okay, and likewise, we then procured FAP147 mutant. Again, we went through the screening, we did studies again, and we showed that, well, while FAP147 mutants do not move, the flagella length is intact. Now, this tells us that therefore FAP147 mutant may not be involved in a certain, attributing the length of the flagella in what we call in our parlance as assembly of the flagella, the length of the flagella, whereas FAP174 might be involved. Of course, we need more studies to confirm this. Michelle and Zhang have used, one particular mutant known as CPC1. I'm showing you CPC1 because CPC1 was also one of the interactors in our immunoprecipitation. Yet again, there is RNA interference, and I'm very, very sure most students are very familiar with RNA interference using the siRNA approach and the miRNA approach, where at the RNA level, you can literally produce, cleave the RNA inhibit the process of conversion of mRNA into protein and therefore a null mutant. Many such mutations have been created even in chlamydomonas, but we tried for hiding. We also procured a FAP75 from the database. Unfortunately, we could not get an RNAi line for hiding. What does it say? It tells us that this approach of RNA interference probably doesn't hold true with hiding, probably. And therefore, this takes us to these approaches of reverse genetics that have several drawbacks. So when we talk of drawbacks, we must also remember that these strategies that you have to use, whichever, tomorrow, even if you have to use Cas9, CRISPR-Cas9, it largely depends on the species and the biological question you are trying to understand, that you're trying to address. Because both these essentially are dependent on the property of the model organism that you're investigating. Sometimes, you would like to introduce the gene by transformation. Transformation may not work with your model organism. It may be as simple a problem as entry of your cassette into the cell. It may be as simple as that. We do not know. It may be as simple as your plasmid which goes inside is getting cleaved off, is getting chewed off, and it cannot render its job. So it could be as simple as these and as complicated as the ones that I've listed here. By and large, whatever they are, I don't expect you to read them or understand or grasp them for now, but the common denominator, the take home from these drawbacks is most of these approaches that I listed and I discussed a few have off-target effects. So when you have an off-target effect, it means this particular approach is not specific. And when it is not specific, you are worried about your favorite protein and the variants of your favorite protein. But for all you know, your phenotype could be because of some off-target effect, some off-target gene that has got manipulated. You don't want that. So that is a background noise. And that is what comes up with this. And here itself, I will tell you how specific is CRISPR-Cas9 versus this. And that is why it becomes a very powerful technique. Some of these approaches are just not possible with complex organisms. People have faced immense problems. And I also spoke about the delivery. So and so on and so forth. Insertions are random. Recombination-based methods are quite inefficient. Selection methods need to be stringent. Sometimes your mutant has to be, characterized with several approaches to literally tell you that this mutant is for good, and you can rely on the phenotype that it actually gives you back, right? And therefore comes the CRISPR-Cas9. So CRISPR-Cas9 story basically started in 1987, where Ishino were trying to study a different gene altogether, an alkaline phosphatase from E. coli. Okay. They sequenced the DNA of the gene, and they also probably were over-ambitious and also sequenced the flanking portions of the gene. To their surprise, they found out something different that was not observed earlier. They found out 5 segments. I've listed them here, 5 segments. I would love you to remember these. 5 segments, roughly 29 bases. These segments are repeats or repeating. And these are palindromes within them. I have shown a palindrome here in one of the consensus sequence of the repeat. Each of these repeats is interspersed with roughly 32 base spacer. So what you have to remember here is Ishino found out flanking sequences in their gene with 29 bases repeating 5 times and spacers in between, each spacer amounting to 32 base pairs. Spacers were different from each other. Okay, it's only the repeats which were identical. I had a consensus sequence. Till around 13 to 15 years, it was Jansen and his co-workers who actually published another paper and trying to understand what Ishino had published. He found out that these particular sequences were actually a cluster, They were regularly interspaced and had short palindromic repeats, and therefore they called them as clustered, regularly interspaced, short palindromic repeats. This is the paper they published. And for short, CRISPR. So this was called as CRISPR sequences. They also found out a little more about these sequences. They found out that associated with these sequences were a collection of some genes which were nearby. They called them CRISPR-associated genes or Cas genes, okay, CRISPR-associated genes. They found out that the genes encoded an enzyme, and not just one enzyme, a few enzymes that could cut DNA, Cas1, Cas2, Cas3, Cas9, Cas13, and so on and so forth. Cas9 is important out here for our technology. And this is what they ultimately showed, that the bacterial genome, the E. coli genome, had what is known as a CRISPR loci with repeats of what are known as CRISPR RNA in gray, spacers in between, some non-coding RNA known as trans CRISPR RNA, because there is slight homology of this trans cRNA, CRISPR RNA with the cRNA, and that is why it's called cRNA, but it is in trans, it is not a part of this cluster. I mean, it is not a part of this loci. Some Cas genes, Cas9 enzyme. This is how Janssen displayed their work. Two other scientists got very curious and they said, let's see why these CRISPR are occurring. So one of the scientists, Eugene Koonin, who's an evolutionary biologist, he quickly understood this. He said that these E. coli are probably using CRISPR as a weapon against viruses. He hypothesized that bacteria use these Cas enzymes to actually grab fragments of DNA or maybe RNA from these viruses and insert them in their genome, so that when there is a subsequent attack, it can use it as a cheat sheet to recognize a subsequent invader, almost like an immunity. And that is why the term adaptive immunity was introduced for this in this particular case. Rodolphe Barangon ultimately tested Koonin's hypothesis. He actually was a microbiologist in a dairy firm. Now he's an associate professor at NC. What he did, he took the milk fermenting microbe Streptococcus thermophilus. He took 2 strains of them. He attacked them with the viruses. He found out that most of the viruses were killed, but most of the bacteria were killed by the viruses, but some of the bacteria he survived. And he found out that those which survived were actually resistant to a subsequent attack by the same virus. So he concluded that there must be a genetic change. He ultimately sequenced and found out that the bacteria actually stuffed DNA fragments from both these viruses as spacers. He called them as spacers. And he said that these spacers remained in the virus and helped the virus to defend the subsequent attack. This is what he said. Subsequently, several scientists, and there are 3 more names that I will come up with a little later. And here I'm trying to show you how the CRISPR-Cas9 was explained and what it does. Let's try to understand this a bit. This is my foreign DNA. This is going to be my target or the spacer. Kindly remember that there is what is known as a PAM sequence, which is known as a protospacer adjacent motif, which is very, very close to this target. The target actually gets decided based on this PAM. So wherever this PAM is, and this is very important for our technology, wherever this PAM is, the target gets decided. The target is roughly around 20 to 29 bases. When such an attack is taking place to a bacteria, it goes through a process known as acquisition. In the step of acquisition, E. coli actually cuts this portion using its Cas enzymes and inserts it in its loci through a process, which is not mentioned out here. And now the bacterial genome will have this new spacer introduced between these repeats using some Cas enzyme. Now, when there is a subsequent attack, what will happen is, this is triggered. This loci is triggered. How is it triggered? Because this primary transcript that is made from this loci is present here. The tra-CR-RNA is present here. These primary transcript and the tra, because I said the tra-CR-RNA has a short sequence that complements the CR-RNA, it aligns with this primary transcript in such a way that in a manner which is known as CR or CRISPR rRNA biogenesis, because it gives rise to CR-RNA. Biogenesis because it gives birth to CR-RNA. This process actually requires RNase 3 and Cas9 from the host. When this complex is happening, that is a ribonucleic acid complex, is ribonucleic RNP complex, the ribonucleoprotein complex, okay, goes and then targets the subsequent spacer which has an identity or similarity to this particular spacer sequence. And because Cas9 is like a double-edged sword, it can actually make double strand mix, you know, with this particular PAM as a as the identifying sequence. This is basically what they discovered, and Baringo and Rudolph and the others get huge credit for, you know, showing us that this is what actually happens in E. coli. Subsequently, many people have tried to understand the sequences and the presence of these in various organisms. And ultimately, there are now two classes of these CRISPR-Cas9 that exist. We will come to some of them a little later. However, type 2 is the one that is present in E. coli and has been the best worked out. And is the Cas9 from E. coli and in some places from Streptococcus that has been used in our technology. So let's try to understand a little bit about the Cas9 enzyme. The Cas9 enzyme basically has 6 domains, the REC1, which is the largest, REC2. So REC1 actually binds to the guide RNA. We'll come to guide RNA a little later. Just give me a minute and I'll tell you what is guide RNA. REC2, whose significance we do not know. Then the histidine, arginine, histidine asparagine portion. This is the nuclease portion. Roof C is again the nuclease. Then a bridge that connects these domains and the PAM interacting domain. So basically, using these activities, 6 domains on this large protein, it can perform several jobs. And what are those jobs? These multiple jobs are actually exploited in the technology. So this is now the fact that we know. It changes the organism's DNA by introducing spacers, the genetic material. Mind you, the genetic material, the spacers can always be removed as well. Okay, and they are passed on. They're passed on from generation to generation. So the progeny of one viral attack which has obtained a spacer gone to the progeny will then be resistant to that virus. Okay, it occurs at the CRISPR loci, and therefore this is the natural adaptive immunity. People have shown that it occurs in nature. More than 60% of bacteria now harbor it, you know, the actual CRISPR contains these 4 simple structures associated with it, which I explained previously. The CRISPR, the Cas9 itself, the system, the types, and type 2 is best studied in E. coli. The guide RNA, now what is guide RNA? Therefore, guide RNA is nothing but the CRISPR RNA, that is CRRNA, and the CRRNA together. So both these, actually, there is a small portion that complements. If you remove the complementary part, this is the complementary part. Okay, that together therefore makes it a guide RNA. And the PAM sequence, which has a canonical sequence. Here it is NGG, specific to Streptococcus pyogenes, and NAG is the one that has been exploited in human cells. And these are the people. These are the 3 people who really get the credit. They developed this very simple cut and paste system, which has now become the genome editing system. Jennifer Doudna, Emmanuel Charpentier, and Virginius Sickness from Louisiana, they were the ones who showed this 3-membered or 3-component system consisting of the enzyme, the CRISPR RNA, and the CRRNA. What they did, They said, let's unite these two because there are complementary portions. They united these two and converted this 3-component system into a 2-component system. They joined the CRRNA and the TRA. Now it becomes a 2-component system. And this is how it works. So if I want to target my gene, I know the sequence. I first search for PAM. I will search for PAM and I go 20 or 29 base pair upstreams and I select this sequence. I make the same sequence and then I join it, ligate it to my CR and TRA RNA, which gives rise to what is known as the guide or the single guide RNA. Now, when this goes to the genomic loci as shown here, with the help of the Cas9, the Cas9 has to go along with it. So SG RNA and the Cas9 now goes to the genome, recognizes, the Cas9 recognizes, and a few base pairs upstream will make a double-stranded break. So this double-stranded break is what you have to remember. Now, depending on the variants of Cas9, which are the applications that are present? Depending on the double-strand break that is made here, you can create an NHEJ because this double-strand break will be recognized by the host in an error-prone NHEJ pathway. Or if you provide a healthy donor DNA, this double-strand break can be repaired and substituted by a new DNA in a process known as homology-directed repair, where there is precise gene editing, provided you give the donor DNA. And in yet another case where you can actually label, you can label the you can label your DNA, you can label your guide DNA. Here, the Cas9 that is modified does not have cleavage activity, so it does not produce double-strand breaks. Both the cleavage activity have been removed, the cleaving activity has been removed. And this can be used for visualization of certain portions of DNA. And these are the applications that are based on the 3 different variants of Cas9. So simply put, very simple, quick, the price is commensurate with the speed, with the accuracy, and also the simplicity. So you can prepare your CRRNA, join it with your TRA RNA, giving rise to your guide RNA. If you want to add a fluorophore, you can add a fluorochrome to it, making a fluorescently labeled guide RNA, join it with a Cas9. If it is labeled, it becomes a labeled RNP, transform your cells, and then ultimately your host will take care of, depending on what you're providing, giving rise to a double-strand break, depending on what you're providing. If you're not providing the donor, it will go into NHEJ. If you're providing the donor, it will go into a precise editing. Therefore, takes us to these application examples listed out here. Modifications shown here in whole cells, single cells, or cell lines, getting a desired phenotype, labeling as I mentioned, and so on and so forth. Therefore, the future of Cas9 is really good. I mean, the past decade has seen a huge use of CRISPR-Cas9 technology in screening targets, in agriculture, ecological purposes, and so on and so forth. Let's come back to CLAMI. So there has been some very good progress in Chlamydomonas, and we were waiting for some time. We were struggling with the system, but now that we know, I'm taking some help from George Whitman for this. I'm going to try to, we've already got the Cas9 and the CRISPR, designed. We've got the constructs, we have the backbone constructs, and we have plans to use CRISPR-Cas9. Unfortunate, with the lockdown, we have not been able to proceed with this work. But once we go back to the lab, we are going to first target specifically FAP75 hiding. So this is where the specificity is. You can create specificity. Specificity is the moot point out here, and that is what I'm trying to drive. It is not random. The Cas9 has a guide RNA, it guides you to the genome, and that's what the specificity that we must remember. And now there are several companies which actually help you with such modules, with such help you, some softwares which also help you. If you have the gene, help you locate your PAN, and so on and so forth. And that is what has become very useful. There are some which actually provide a suite, for doing your CRISPR. So this is fairly advanced now. You can almost, I mean, you can do it in almost any organism, I guess. We are going to try it with chlamydomonas. People in the field have been successful. In fact, there is one that has been successful with flagellar gene also from Professor Whitman's lab. So with this, I thank once again for your kind attention, and I thank the organizers once again, in particular Dr. Saraf, for inviting me for this really wonderful platform where I can share not only my research program but also the superb technology of CRISPR-Cas9. Thank you very much.
Thank you, madam. I would like to express our appreciation of your most interesting talk. And on behalf of everyone here, I would like to thank you for your presentation. And I'm sure after the stimulating presentation, the delegates would like to ask you questions.
Sure.
And yeah, madam, we have created a WhatsApp Telegram group for participants.
Okay.
So they are encouraged to post their queries on the group.
Sure,
And
sure.
we'll get back to you with those, we'll email to you all those queries, madam.
No problem.
Yeah, and I'm sure you will get back to us with that.
Yeah, thank you.
Yeah.
Thank you so much, Dr. Saraf.
Thank you, madam. Thank you.
Thank you. Stay safe, stay healthy. Bye-bye.
Bye. Thank you, ma'am. And with this, we come to end of this session. And now we are breaking for lunch. At 3 o'clock, we are starting with our next session. So see you at 3 o'clock. Thank you. Thank you, everyone. Thank you for joining us for this session. Thank you, Amol sir.